Bismillahirrahmanirrahim. Assalamu alaikum wa rahmatullah. Rabbi shrah li sadri wa yassir li amri wahlul 'uqdatam min lisani yafqahu qawli. আমরা এখন introduction to systemic bacteriology এটা নিয়ে ছোট একটি আলোচনা করব ইনশাআল্লাহ। এই ছোট লেকচারটির লেসন প্ল্যান নিচের মতো। আমরা meaning of bacteria, distribution of bacteria with their numbers, number of bacteria in the human body, principle and steps of laboratory diagnosis of a bacterial disease, and classification of bacteria. The word bacteria is the plural of the new Latin bacteria, which is the Latinization of the Greek bacterium. That means a staff or a cane, because the first bacteria to be discovered were rod-shaped. Microbes, that is bacteria, are everywhere. Everywhere means in the sea, deep sea, in the ice, in the garden, in the volcanoes, in the deserts, and everywhere in the food. Bacteria are everywhere. The number of bacteria in this earth is 5 million trillion. I repeat, 5 million trillion. That is, after 5, 30 zeros after it. 5 to the power 35 into 10 to the power 30 zero. And University, Mr. Dr. Whitman from University of Georgia, USA, he said, there simply hadn't been any estimates of the number of bacteria on Earth, because they are so diverse and important. We thought it made sense to get a picture of their magnitude. So, distribution of microorganisms, as we have seen in the air, in the soil, in the water, in the animals, and in the human body. There are typically 40 million bacterial cells in a gram of soil. 40 millions means 4 crores. 1 million bacterial cells are present in 1 ml of fresh water. In all, there are approximately 5 nonillion, that is 5 million trillion, 5 into 10 to the power 30 bacteria on the earth, forming a biomass that exceeds that of all plants and animals. সমস্ত প্লান্ট এবং অ্যানিম্যাল মিলে যত ওজন হবে তার চেয়ে বেশি হলো ব্যাকটেরিয়ার ওজনের সমষ্টি। Most bacteria have not been characterized, and only about half of the phyla of bacteria can be grown in the laboratory. There are approximately 10 times as many bacterial cells in the human flora as there are human cells in the body, with large numbers of the bacteria on the skin and as gut flora. অর্থাৎ আমাদের নিজেদের যতগুলো সেল আছে তার চেয়ে 10 গুণ বেশি হলো ব্যাকটেরিয়াল সেল, 10 times. Vast majority of the bacteria in the body are rendered harmless by the protective effects of the immune system, and a few are beneficial. And the beneficial bacteria are known as probiotics. However, a few species of bacteria are pathogenic, and they cause infectious disease, which include cholera, tuberculosis, leprosy, syphilis, and so many to be named. Let us discuss about the principle of laboratory diagnosis of a bacterial disease. The principle is based on four things. First, demonstration of the causative agent by microscopy. That microscopy may be with staining or without staining. If with staining, staining may be of gram staining, A-based gel-Nansen staining, fluorescence staining, and microscopy can be also light microscopy, dark ground microscopy, and different types of microscopy. Second thing is isolation and identification by culture. Many of the pathogenic bacteria can be cultured in artificial media, and few cannot be cultured. Other than culture, there are immunological tests for detection of antibody or antigen by immunological test. Mostly antibodies are detected from the blood and antigens are detected from the body fluids. And newer technology, nucleic acid-based technologies, also known as NAT or PCR, like this. And in special cases, for some bacteria, we need some special test, like for diphtheria diagnosis, we need toxigenicity test. Now, based on this principle, what are the steps of laboratory diagnosis of a bacterial disease? First step is you need a specimen, appropriate specimen, and usually that depends on the site of the infection. If there is pneumonia, you will have to take sputum. If septicemia, blood. If diarrhea, stool. If urinary tract infection, urine. And if in case of wound infection, you will have to take the swab from the wound. Then, as we have said in the previous slide, first step is we will have to see whether microscopy can be done. A microscopy again, unstained or stained, dark ground microscopy, immunofluorescence microscopy, or light microscopy. And when stained, depending on the nature of the bacteria, gram staining, gel-Nansen staining, or some other special staining can be done. Next to microscopy, we will have to look for isolation and identification, commonly known as culture in artificial media. For different types of bacteria, different types of media, incubation time, temperature, and environment varies. And one advantage of this culture technique is that sensitivity, in vitro sensitivity can be done, sensitivity to different types of antibiotics. There are manual methods as well as automated methods, disk diffusion as well as MIC methods. For example, for pulmonary tuberculosis, sputum is to be cultured in a special media like Lowenstein-Janssen media, is to be incubated at 37 degrees centigrade aerobically for 4 to 6 weeks. But for ordinary bacteria, mostly it is overnight or it is 24 to 48 hours. Immunological tests include immunochromatographic test, very popular nowadays, enzyme-linked immunosorbent assay, or some other immunological methods for detection of antibody as well as antigen. And nucleic acid-based techniques, also known NAT, nucleic acid amplification test, PCR, polymerase chain reaction, nowadays very common and popular method. Classification of bacteria. Hans Christian Gram in 1884, he discovered the staining method. By that staining method, and based on the shape and size of the bacteria, we can classify the bacteria in two broad groups: gram-positive bacteria and gram-negative bacteria. Gram-positive bacteria may be again rod and cocci, and based on their oxygen requirement, spore formation, there are many classifications. Gram-negative bacteria also there are, and we will go to a simpler method, say gram-positive bacteria, cocci and bacilli. Cocci, those which are round in clusters or tetrads, Staphylococcus, Micrococcus, and Peptococcus, of which Staphylococcus aureus is very important. Oval shape and in chains, mostly they are Streptococcus, Peptostreptococcus, and Enterococcus. All are common. Bacilli may be filamentous, spore-bearing, or club-shaped. When they are filamentous, maybe extensive branching or branching rudimentary or absent. When there is extensive branching, actinomyces or nocardia, and branching is rudimentary or absent, lactobacillus, Eubacterium, erysipelothrips. Spore-bearing gram-positive bacilli, they constitute a very important part. Bacillus anthracis, Clostridium tetani, perfringens, etc. And the club-shaped bacteria, Corynebacterium, Listeria, Mycobacterium, these bacteria important gram-positive, club-shaped or in palisates. Now, gram-negative bacteria. Gram-negative again, cocci and bacilli. Cocci only one group, Neisseria and Veronella. Most of the bacteria are gram-negative bacilli. Bacilli again, based on the size, if medium-sized coccobacilli, they are Acinetobacter or Moraxella. Teeny coccobacilli, Brucella, Bordetella, and Bacteroides. Pleomorphic coccobacillary and filamentous. In this group, Haemophilus, Bordetella, Pastorella, these are important members. Some bacteria have exaggerated pointed ends, and some have coiled sphaeroplastic. These are Fusobacterium nucleatum and Streptobacillus. In this group, gram-negative bacilli, there are some carved comma-shaped bacilli, most important, Vibrio, Campylobacter, and Helicobacter. And rest, last one, uniformly bacillary state. They are again Enterobacteriaceae, a large family of bacteria, and many pathogens are there. Pseudomonas, important for hospital-acquired infection. Aeromonas, also a cause of diarrhea legend. Aeromonas, Leishmaniasis like this. So we came to an end. We will just summarize what we told in this lecture. We can say take-home message. First of all, bacteria present everywhere in the earth. Number of bacteria in the earth is 5 million trillion or 5 nonillion. That is 5 into 10 to the power 30. Human body contains 10 times as many bacterial cells as resident flora than the number of human cells in the body. Principle of laboratory diagnosis of bacterial disease is based on microscopy, isolation, identification, immunological test, and nucleic acid-based test. For some bacteria, all these tests can be done, and for some bacteria, microscopic demonstration is enough. Many bacteria can be isolated and in culture, and some bacteria cannot be isolated. Some bacteria requires longer time for isolation, for example, Mycobacterium tuberculosis. Nowadays, immunological tests are available for most of the bacteria for detection of antibody or antigen, and nucleic acid-based test is nowadays very popular, replacing the culture techniques. But culture technique is still very important because sensitivity can be done after isolation and identification of the bacteria. You have seen bacteria can be classified on the basis of gram staining, their shape, size, oxygen requirement, spore formation, etc. Thank you for watching, and hope if you have any query, please mention in the comments. I will answer. Thank you very much. Assalamu alaikum wa rahmatullah.